Thank you for the opportunity to speak to you today. My talk is entitled, Will SARS-CoV-2 become resistant to the current vaccines? Implications for vaccine development, though it's possible that the answer to this has become clearer in the last few days. So I'll leave you to judge that for yourself. So we all know from many studies now that SARS-CoV-2 infected individuals develop neutralizing antibodies and that these can persist for many months. Neutralizing antibodies are also considered the primary correlate of protection against reinfection for most vaccines, and this includes SARS-CoV-2. But it's important to note that most of the vaccines that have been made thus far have been designed on the original Wuhan 1, or in some cases D614G strain, and that most of the assessments that have been made thus far have been based also on these two strains. SARS-CoV-2 monoclonal antibodies are also being pursued as therapeutics, and so there's great interest in these types of antibodies. But in the backdrop to this, one of the issues that has really come to the fore in the last few weeks is that SARS-CoV-2 spike is mutating. So the SARS-CoV-2 spike has really always been mutating. Mutations occur regularly during the viral replicative cycle, and they can be seeded through superspreader events, or they can be selected for. Most of the changes that occur during natural replication do not confer a selective advantage to the virus, and they do not end up replacing pre-existing lineages. But this is not always the case. And in fact, we've already seen one clear example where a mutation that has emerged in SARS-CoV-2 has given the virus a clear selective advantage and has resulted in global replacement. And that's the emergence of the D614G mutant. This mutation, which arose some months ago, has now replaced essentially all the previously circulating viruses and now dominates global sequences. And this can be seen here in this image from a very elegant paper from Betty Korber and colleagues. The issue of variants of concern, which the name for variants of concern is obvious, is because these variants, these mutants that may come to dominate, can have several impacts on the virus that have public health implications. These mutations or suites of mutations in some cases may result in increased disease severity, and some of these reports have come out of the UK in recent weeks. They may result in the virus becoming much more transmissible, and there's several examples of that. And more recently, the focus has been on the possibility that these variants of concern may evade the immune response, and that's really going to be the focus of my talk for much of the next few slides. So 3 major variants of concern have been identified based initially on where they were first described. There is the 501YV1 variant or lineage, otherwise known as B117. There's the 501YV2 or B.1.351, and then there's the 501V3. 501YV1 was first identified in the UK, V2 in South Africa, and V3 or P1 as it's otherwise known, identified first in Brazil. And these maps here from the Pangolin new variant report show very clearly that these variants have now started spreading across the world. And the dark green here shows community transmission and the light green shows imported transmission. But you can see that these variants are no longer limited to the countries in which they were first described. They have also been shown to have the mutations have been shown to have many different impacts on the virus. The variant that was first described in the UK has been clearly shown here to have increased transmissibility. And more recent reports suggest that the 501YV1 is also associated with increased disease severity. In the case of 501YV2, which I'm going to speak about much more in this talk, there's some evidence, though it's not entirely clear, of enhanced transmissibility and increased evidence, which I'll show you in this talk and which has been described elsewhere, of immune evasion. In the case of P1, also some evidence of immune evasion, and I'll come back to that. But I think what's important to note here is that there is some degree, at least, of evidence emerging of descriptions of convergent evolution, in the sense that all of these 3 variants of concern all have an N501Y mutation, which is the particular mutation that has been associated with both enhanced transmissibility and perhaps disease severity. Our interest in new variants that are emerging in SARS-CoV-2 really began with the detection of the 501YV2 lineage in South Africa. And this graph here shows you the emergence and subsequent dominance, really, of 501YV2. This is a paper from Tulio D'Oliveira and colleagues. And in yellow here at the end, you can see the bar chart show you the dominance of the 501YV2 variant or lineage in South Africa. And you can see clearly that by the end of November, that lineage was responsible for almost all new infections in South Africa. And now this continues to be the case. Sequencing efforts in South Africa suggest that in excess of 90 and perhaps 95% of new infections are caused by the 501YV2 lineage in South Africa. And of course, as I mentioned, it's no longer limited to South Africa. The 501YV2 lineage is characterized by multiple mutations, 9 mutations just in the spike. And I'm going to focus on the spike largely, as I mentioned, because it is the target of SARS-CoV-2 vaccine design and is the most frequently targeted, in fact, the only target of neutralizing antibodies in SARS-CoV-2. The mutations that characterize 501YV2 are really limited to 2 areas, and that immediately gives you a clue that these are immune evasion mutations. Up here at the top, so this here is the SARS-CoV-2 spike, a model of the SARS-CoV-2 spike. And up here at the top in yellow is the receptor binding domain. This is the part of the spike that interacts with the ACE2 receptor, but it's also a dominant target for neutralizing antibodies, as I'll show you on the next few slides. And there are 3 mutations within this region, 501, which I've already mentioned, but also mutations at positions 417 and 484. The other cluster of mutations that define 501YV2 are located within the N-terminal domain, and that's this area shown here in cyan. And you can see that there's several mutations in that region, positions 18, 80, 215, 246, and also a 3 amino acid deletion at positions 242 and through to 244. The clustering of these mutations, with the exception of this mutation that sits down here at position 701, as I mentioned, is indicative of immune escape. Whenever you see clustering of mutations together in a 3-dimensional space, this is of concern to virologists. But the main reason that we are concerned about these clusters of mutations is that both the RBD and the NTD are the major targets of the SARS-CoV-2 neutralizing antibody response. This is particularly true of the RBD, and the neutralizing response is dominated by RBD antibodies. There are many antibodies to other regions of the spike, but when you think particularly about the neutralizing antibody response, it's dominated by RBD antibodies. And in fact, most MAB antibodies, monoclonal antibody epitopes, overlap with the ACE2 binding site. And I'm going to speak much more about the class 1 monoclonal antibodies and the class 2 monoclonal antibodies in the next few slides. The N-terminal domain is perhaps somewhat less frequently a target of neutralizing antibodies, but certainly there are many examples in the literature of monoclonal antibodies that have been isolated to this region. And these include 4A8, which again, I will speak about in the next couple of slides. So the way we approach the question of whether these clusters of mutations in 501YV2 are associated with immune evasion is by using a pseudovirus neutralization assay. This assay uses 293 T cells that overexpress ACE2 as a target cell line. These cells were very kindly provided by Mike Farzan. And we used three variants. We used the variant that was originally circulating in South Africa, which is called the wild type, which has just the 614G mutation. We also constructed, given the huge interest in the receptor binding domain as a target of neutralizing antibody, we also constructed an RBD triple mutant. And this mutant here contains only the mutations of positions 417, 484, and 501. And then we also constructed the full 501YV2 variant, which contains all the mutations that I showed you on the previous slide. So all 3 of these were used in pseudovirus neutralization assays, and they're the basis of the data that I'll be showing you next. We began by assessing the sensitivity of 501YV2 to monoclonal antibodies. It's a nice, clean way of assessing particular sites. We started with the class 1 monoclonal antibodies, and structural analyses and modeling have indicated that these monoclonal antibodies are heavily dependent on the site at position 417, which, as I've already mentioned, is one of the sites that is mutated in 501YB2. To understand whether these monoclonal antibodies, these class 1 monoclonal antibodies, were impacted in the new, whether these monoclonal antibodies were still capable of neutralizing 501YB2, we performed both ELISA and neutralization assays. So we selected 3 representative monoclonal antibodies shown up here at the top, and we ran them in ELISA, that's the top panel, and neutralization assays in the bottom panel. The key is down at the bottom, but if you look first at the ELISA data, the black lines indicate the binding of these monoclonal antibodies to the wild type D614G. And down here at the bottom, you can see the binding of these monoclonal antibodies, or the lack of binding, in fact, to 501YV2. So you can see binding of all 3 monoclonal antibodies is completely knocked out. Down here at the bottom, you can see the same for neutralization. Again, in black is the neutralization of the wild type. And in red is 501YV2. And down here at the bottom in yellow is the triple mutant that contains only the RBD mutations. And you can see that in both 501YV2 and the RBD triple mutant, there is no neutralization whatsoever by class 1 monoclonal antibodies. This is the same set of data for the class 2 monoclonal antibodies. These monoclonal antibodies are heavily dependent on the position at 484. This is the same experiment using 3 representative class 2 monoclonal antibodies. Again, ELISA up here at the top, and you can see that there's no longer any binding whatsoever to 501YV2. And down here at the bottom is neutralization data showing, again, the same thing, fairly potent neutralization, in some cases, very potent neutralization of the D614G, the original variant that was circulating in South Africa. But you can see that there's no longer neutralization of either the RBD triple mutant or the 501YV2. We also looked at an NTD-directed monoclonal antibody. This is, I mentioned 4A8 on my introductory slides. An analysis of several of the NTD-directed monoclonal antibodies shows that they're heavily dependent on this N5 loop supersite shown here, as well as that position 246, which is mutated in 501YV2. You can see that when you extend this, this is a model using 4A8, but when you extend this to many other monoclonal antibodies targeting the NTD, you can see that all of them are dependent on, to some degree, on the N5 loop supersite. In 501YV2, there's substantial remodeling of the NTD domain from an antigenic perspective. You can see here in 501YV2 that the N5 loop is shifted substantially. And there's also a clash here that results from the mutation at position 246 in 501YV2. And this would suggest that, NTD-directed monoclonal antibodies may no longer be able to recognize 501YV2. And indeed, when we perform ELISA and neutralization assays, we see that 4A8 is no longer able to bind to 501YV2 nor to neutralize. So altogether, the monoclonal antibody data that I've shown you shows that 501YV2 is completely resistant to 3 classes of therapeutically relevant monoclonal antibodies. Though important to point out that there are a few other monoclonal antibodies that are likely to be unaffected by these mutations that are therapeutically relevant. So up to now, I've been focusing on neutralization by monoclonal antibodies. Perhaps of more relevance from a public health perspective is the capacity of convalescent plasma from people previously infected with the original variant to be able to neutralize the 501YV2. And that's the next question we asked. We selected 44 donor plasma or sera that were obtained from people previously infected with the D614G variant, which was the dominant variant in South Africa. We very deliberately selected sera with a wide range of titers. In gray here, you can see, this is a logarithmic scale. In gray here, you can see high titer individuals with titers in excess of 1 in 400. Down here at the bottom, you can see that half of the sera we chose had lower titers, somewhere between 1 in 50 and 1 in 400. And the limit of detection for this assay is 1 in 20. So this is the neutralization capacity against the original variant. What happens when we test against the mutants? When we test against the RBD only pseudovirus, so this contains only 3 mutations, you can see a 5-fold reduction in mean titer if you compare the 614G variant to the triple variant. And perhaps more striking, 27% of donors are no longer able to neutralize the pseudovirus that contains just these 3 mutations. That effect is even more pronounced when you test neutralization of these plasma against 501YV2, which contains the full suite of mutations. Here we see a 13-fold reduction in mean titer against 501YV2, and 48% of donors no longer show any neutralization whatsoever. Important also to point out that this is very much a titer-dependent effect. So if you look in the light gray, you can see that individuals who started with a very high titer against 614G generally retain some activity against 501YV2, whereas individuals down here with titers somewhere between 100 and 400 lose much of their activity against 501YV2. And very important to point out that this is probably the most relevant from a sort of a reinfection point of view, because most individuals who become infected with SARS-CoV-2 show titers in the lower range rather than these individuals up here who are hospitalized folks and have much higher titers. So there's clear evidence, I think, from our data and from other studies that show that the South African variant does exhibit substantial levels of immune escape from neutralizing antibodies. But I think what's probably even more striking is the fact that this is not a unique escape pathway for the South African variant. The South African variant 501YV2 shares major RBD mutations with P1, which is the variant first described in Brazil. Both of these contain mutations at 4 and 7. It's a slightly different mutation at 4 and 7, but mutations at 4 and 7, and 484 as well as 501. And what's perhaps even more striking is that even in the 501YV1 variant, which was first described in the UK, at low levels for now, people are also detecting the 484 mutation. So I think that speaks to the extremely strong pressure on the virus to escape from neutralizing antibodies, and the fact that most people have a very, very similar and focused response to SARS-CoV-2 infection, which makes it in some ways much easier for the viruses across the world to converge on the same escape mutation. So what does this all mean for vaccines? Much of what I've shown you, everything I've shown you so far is monoclonal antibody data and convalescent plasma. But what happens for vaccines? Here I'm showing you published data investigating the effect of sera obtained from vaccinees, either from Moderna or Pfizer, the 2 mRNA platforms, and asking how sensitive both the variant detected in the UK, defined here as UK Delta 8, and 501YV2, defined here as SA Delta 9, are to antibodies in people who received the Moderna and the Pfizer vaccines. So you can see here in black is the sensitivity of the original variants to sera from Moderna and Pfizer. There's a slight decrease, somewhere between around about 2-fold for the sera to the UK variant, 1.8-fold for Moderna and 2-fold for Pfizer. But as you can see from the substantially bigger drops, there's a much more pronounced effect against the South African variant, 8-fold for Moderna and nearly 7-fold for Pfizer. The same is true, and this is data in a preprint led by Shabir Mahdi. This is data from our lab investigating the impact of the 501YV2 variant on vaccinee sera from samples from the AstraZeneca trial that was conducted in South Africa. These are the same constructs that I've showed you in previous slides. And you can see that there's a range of titers here against the original variant. Tested against the RBD only variant, you can see substantially lower titers. And against 501YV2, nearly 78% of vaccinees no longer show any activity against 501YV2. So really dramatic drop-offs against 501YV2. This is supported by evidence now from very recent evidence now from 3 vaccine trials that were conducted in South Africa. This is the Johnson & Johnson trial over here, Novavax, and the AstraZeneca data. The AstraZeneca data comes from a very small trial that was not particularly powered to pick up efficacy against the new variant, but nonetheless is instructive. When you look at efficacy of all of these 3 vaccines in the US, the UK, and Brazil, you can see that efficacy is fairly high, somewhere between 70 and 90%. However, if you look at efficacy specifically within South Africa, you can see a dramatic drop-off. Both the Johnson & Johnson vaccines and the Novavax vaccines decline in efficacy to somewhere around 50 to 60%. And the AstraZeneca vaccine shows very low efficacy against South African strains. And the reason that these trials are able to pick these up is that these trials in South Africa happen to be performed at exactly the moment that the new variant was starting to dominate in South Africa. This means that the investigators were able to assess the breakthrough infections by sequencing them, and were able to show that in excess of 90% of breakthrough infections in all 3 trials were 501YV2, further evidence of reduced efficacy against these particular variants. So I think the data is very suggestive that the lower vaccine efficacy observed in South Africa is likely due to the dominance of 501YV2. However, very important to point out that what is less clear is the efficacy of these vaccines against in preventing severe illness in people, even those infected with 501YV2. The data that we've heard about so far from Johnson & Johnson indicates that this vaccine still retains its ability to protect from severe infection, but we don't know anything about the ability of the Novavax or AstraZeneca trials to prevent severe illness. And of course, from a public health perspective, preventing severe illness is the #1 priority. So that is something that needs a lot of further study to understand whether this is true, and if so, what the mechanism is for preventing severe illness in people who become infected. So in conclusion, I think that the data I've shown you shows very clearly that the 501YV2 lineage exhibits complete escape from 3 classes of therapeutically relevant monoclonal antibodies. In South Africa, we're unlikely to receive those monoclonal antibodies anytime soon, but I've already shown you that these, the 501YV2 lineage or variant is no longer limited to South Africa. 501YV2 also shows substantial or complete escape from neutralizing antibodies in COVID-19 convalescent plasma. And that has serious implications for the next waves of virus that come through. What I didn't show you is data that we have that shows that a significant proportion of RBD binding antibodies still remain reactive against 501YV2. And that's an important finding, because that does suggest that together with cellular immunity, these RBD binding antibodies, which may be mediating FC effector functions, for example, may account for the retention of the protection from disease in trials, despite the fact that there's lower activity of neutralizing antibodies. Overall, I think these data highlight the prospect of reinfection with antigenically distinct variants. And in fact, in South Africa, we're already seeing that occurring, and does provide a mechanism for the reduced efficacy of current spike-based vaccines that I showed you in the last slide. So where do these variants come from? I think the selection, the convergent evolution towards mutations that confer neutralization escape suggests that ongoing transmission and some level of seropositivity, in some places incredibly high. For example, in South Africa, we have regions where there's incredibly high levels of population seropositivity. And this likely selects for neutralization resistant variants. I think an unknown question that many of us are scrambling to answer is, how will the immune system see these new variants? Will the titers against the new variant be as high as against the old variant? And will there be cross-reactivity in people who are infected with the new variants? Will their plasma be better able to recognize not only the new variant, but also the old variant? In terms of how this data and similar data impacts on vaccine design, many of the major vaccine developers are already scrambling to incorporate new variants in their platforms. A major question here will be whether vaccination, again with vaccines that incorporate these new variants, will result in some level of cross-reactivity. And if not, this may present a short-term solution. It's certain, I think, that SARS Cov-2 will continue to mutate. And the issue is whether the incorporation of these new variants may result in vaccines that are able to confer some level of cross-reactivity, or whether we're looking at a situation similar to that of flu vaccines, where we may need to replace the inserts of vaccines more frequently than we had hoped. Clearly, there are many lineages of SARS-CoV-2 emerging globally, and often these have convergent evolution, again, a focused immune system resulting in a focused escape pathway. And tracking these will continue to be critical for public health. And with that, I will end. I'd like to thank particularly Kurt Wibner, who led the studies in our lab of convalescent plasma and monoclonal antibodies, but many people in our lab who really worked through the Christmas break in South Africa after the 501YV2 variant was identified and worked hard to understand what the phenotypic significance of this was. Thank you to our collaborators at the South African National Blood Service, the University of Pretoria, University of KwaZulu-Natal and Wits University, and the 501YV2 consortium of South African scientists chaired by Drs. Hanekom and D'Oliveira. Also like to thank our funders, largely the South African Medical Research Council, who's funded most of this research, and our wonderful lab here who works incredibly hard to support these studies. Thank you.